Our discussion for today will be Biology of the Cell, Chapter 4 of the McKinley text. So right off the bat, we're going to talk about a few different things that you want to make sure you can actually accomplish after going over this first section. You should be able to distinguish among the light microscopy, scanning electron microscopy, and transmission electron microscopy. You should be able to describe the range and the size of human cells, to at least to some extent. Identify some of the shapes and cells may exhibit, some as opposed to remembering every single one of them necessarily. Describing the main structural features of the cell from a general standpoint, you should be able to do that. Identify the membrane-bound and non-membrane-bound organelles, and distinguish between organelles and cell inclusions, let alone explain the general functions that cells must perform. So cytology is simply the study of cells. Microscopes are necessary to do it because they're obviously measured in micrometers, extremely small, too small for the naked eye, which is one micrometer is equivalent to 1/10,000th of a centimeter. Microscopy is using a microscope to view small-scale structures and using staining techniques to provide contrast so you can see them, which I'm sure you've done in both high school and college level biology and some chemistry. That being said, light microscopes, LM produces a two-dimensional image and it passes light through a specimen. Electron microscope is a beam of electrons which illuminates the specimen. It has a greater magnification and resolution than a light microscope. The two different variations of electron microscope are the transmission electron microscope, which directs an electron beam through thin-cut sections to get 2D images, whereas a scanning electron microscope directs that beam across the surface of a specimen to get you 3D images. And here you have that light microscopy and the scanning electron microscopy and then the transmission electron microscopy, clearly giving a much more clear picture as you move through those different forms of microscopy. For cell size and shape, cells certainly vary greatly in their size and their shape, not just between cells of different types, but within people to some extent. That said, an erythrocyte or red blood cell is between 7 to 8 micrometers, whereas a human oocyte or ovarian cell has a diameter of about 120 micrometers. Most are microscopic, most cells. So shapes can vary to spherical, cube-like, column-like, cylindrical, disc-shaped, or irregular. And you can see a little comparison. It'll come back to when needed to be able to get a better idea of like the sizes of some of these different cells. A human oocyte there, and the red blood cell there, and then a mitochondria, of course, being smaller than that. Going to viruses, ribosomes, large macromolecules, small molecules, and of course down to an atom. And the variety of cell shapes, generally speaking, an irregular shape like nerve cell, the biconcave disc like a red blood cell, like an erythrocyte, a cube shape like the kidney tubule cells, column shape like intestinal lining cells, and a spherical like cartilage shell cells. This kind of throws you off with the cube shape here, but basically cartilage type cells, these spherical type cells here, and cylindrical like skeletal muscle cells. As far as the plasma membrane, it forms the outer limiting barrier separating the internal contents from the external environment, separating the extracellular fluid from the intracellular fluid, and all the contents of the intracellular fluid, let alone all the contents outside the cell. There are modified extensions of the plasma membrane, the cilia, which is used for motion and some sensing, the flagellum for motion, particularly like the sperm, microvilli for absorption and secretion of chemicals. The nucleus is in the center of the cell, enclosed by a nuclear envelope. It contains genetic material. DNA, of course, is the basis for growth and development of the cell, and the nucleoplasm is the inner fluid. The cytoplasm contains all the cell contents between the plasma membrane and the nucleus. Essentially, it's why we call it non-nuclear. It includes the cytosol, the organelles, and all the inclusions. As far as the cytoplasmic components, in the cytosol, the intracellular fluid, it's a viscous fluid of cytoplasm, has a high water content, contains dissolved macromolecules and ions. We'll talk more about that later on. The organelles are little organs inside that cell, inside the cytoplasm. They're complex organized structures within those cells, unique shapes and functions. You have the membrane-bound organelles and non-membrane-bound organelles. So the membrane-bound organelles are enclosed by a membrane. They separate contents from the cytosol, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, peroxisomes, and mitochondria. We'll talk all about all those, at least to some extent, as this whole section goes forward. The non-membrane-bound organelles, they're not enclosed within a membrane. They're composed of proteins, ribosomes, cytoskeleton, centrosome, and proteasomes. Inclusions are basically stored temporarily in the cytosol. They don't stay permanently. They're not considered organelles. They're molecules added to and removed from continuously. Some pigments like melanin, glycogen, triglycerides, and a few others. So here we can get a good little picture of the cell. I'll zoom in for you to be able to go back and look at it, whatnot, and reference it as necessary to be able to see all the various components that go with the cell, to be able to quiz yourself and check on all the different aspects of the components of that cell. I'm trying to give you a good little picture to be able to go back and look and quiz yourself as necessary. As far as common features and general functions, cells performs three basic general functions, basically maintaining the integrity and shape of the cell, dependent upon the plasma membrane and internal contents, of course, obtain nutrients and form chemical building blocks. Obviously, they're trying to harvest energy for survival, their survival, let alone the entire organ and the organism and so on. Disposal of waste, avoiding accumulation that could disrupt cellular activities. And some are capable of cell division as well, where you make more of the same cell type, right? It helps maintain tissue by providing cells for new growth and replacing dead cells. So you should be able to go back to that section and now be able to note which cell is larger, an erythrocyte or a human oocyte, what are their respective sizes, the various sizes, erythrocyte being, of course, 7 to 8 micrometers and oocyte being about 120 micrometers. The structural features of a cell and what side of the structure is responsible for forming the boundary of a cell and maintaining its integrity. That should be pretty straightforward, but go back as you need to. For section 4.2, lipid components, a plasma membrane. The fluid mixture, it contains basically the fluid mixture composed of equal parts lipid and protein by weight. It regulates movement of most substances in and out of the cell to the extracellular fluid outside the cell and to intracellular fluid inside the cell. And it contains several different types of lipids, phospholipids, cholesterol, and glycolipids. Phospholipids, it's often called the balloon with two tails, right? All those tons of balloons of blue tails forming the whole phospholipid bilayer. The hydrophilic head and the hydrophobic tails. They form two parallel sheets, and that bilayer with the polar heads and the non-polar tails in between, something that might be able to pass through a polar barrier would have difficulty with the non-polar barrier. Something with a non-polar barrier would have difficulty passing through the polar barrier. Therefore, it's easier for it to keep contents within the cell that should stay inside the cell and contents outside the cell that should stay outside the contents of the cell, with a few exceptions to what is easy, some smaller structures that can penetrate those layers, and we'll talk more about that. Cholesterol, other lipid components, it's scattered within the phospholipid bilayer, strengthens the membrane. It stabilizes the membrane against temperature extremes. The glycolipids are lipids with attached carbohydrate groups, or glyco, right? They're on the outer phospholipid region only, and they help form the glycocalyx, okay, along with a lot of other substances. Here we'll get a better picture of this here. Also for pretty good reference here, you'll see that phospholipid bilayer, see the glycolipids there, refer to the carbohydrate here, portion off here, interstitial fluid all outside the cell here, and of course all the intracellular fluid here in the cytosol. You can see an integral protein here and a peripheral protein, filaments of the cytoskeleton here, and better examine another protein here. We'll talk a lot of these proteins here in a little bit. And of course, you want to make sure you understand that this plasma membrane provides a physical barrier, a selectively permeable boundary. Some things get across easier, like sodium has a tendency to break the gum internally fairly easily and leak through. Electrochemical gradient, establishing that maintain that charge that typically speaking, we look at an electron charge or negatively charged for the internal parts of the cell, which naturally helps you understand that the outside is going to be more slightly positive, but the more important focus point is that the internal is negative. And of course, communication or establishing it, so those receptors that recognize and respond to different molecular signals. The membrane proteins, they compose half the plasma membrane by weight. They float and move within the fluid bilayer, and they perform most of the membrane's functions. And the two of those different types of membrane proteins are integral and peripheral. And that said, the integral is within and extending across the phospholipid bilayer, with the hydrophobic regions interacting with the hydrophobic interior, and the same with hydrophilic. And many of those are glycoproteins with the carbohydrate portion, those integral proteins. And the peripheral proteins aren't embedded in the lipid bilayer. They're loosely attached to the external or internal surfaces of the membrane. As far as the categories of proteins, transport proteins regulate the movement of substances across the membrane, as the name would suggest. Provides channels, carriers, and pumps, symporters and antiporters. We'll talk a lot about this in a later section of the same chapter here. Cell surface receptors, they bind molecules called ligands. Neurotransmitters are released from a nerve cell that binds to a muscle cell to initiate contraction. That's significant. We'll come back to that too. Identity markers communicate to other cells they belong to the body. Those markers are used to distinguish healthy cells from the cells that need to be destroyed. And of course, you have enzymes that can be attached to either internal or external surfaces. And of course, enzymes, as always, catalyze chemical reactions. And as so often said, when we talk about enzymes not doing their job, it slows everything else substantially. Now, that slowing could potentially cause some serious damage and create possibly some structures could potentially die, because of the slowness. But by itself, lacking enzyme does not mean something is not going to happen at all. It just means it greatly slows the process, so it greatly decreases probability of things working like they should when enzymes are not present. Anchoring sites, their job is, of course, to secure the cytoskeleton to the plasma membrane, and cell adhesion proteins perform cell-to-cell attachments. And here we can see the plasma membrane proteins, the various types. You have the channel, showing from the interstitial fluid outside the cell, the cytosol inside the cell. And typically speaking, in a channel, typically is moving from the external cellular fluid to the internal cellular fluid. Transport proteins going to get crossed by a carrier protein. Pump, like the sodium-potassium pump, was a very significant one we'll talk a little about. Cell surface receptors, where ligands are attached here. Identity markers with glycoproteins and glycolipids, whatnot. And of course, enzyme with the substrate the product help form there at that particular location. And then we have the anchoring site, where basically it's attached with the cytoskeleton protein, and of course the cell adhesion proteins as well. So you should be able to now look back and note what lipids maintain the physical barrier of the plasma membrane, what type of plasma membrane proteins provide the means for moving materials across the plasma membrane, and what are the three subtypes. And we'll come back to section 3 shortly.